So positron emission tomography, or PET for short, is a functional neuroimaging technique that was invented in 1953. So of the two hemodynamic techniques that we're going to talk about, this is the older one. So let's start by watching this brief video from Imperial College London. It has really nice visuals that explain the basic physics of PET.
Meet Dave. He's about to have a brain scan. Using the latest PET imaging technology, doctors will see Dave's brain at work. It's all possible because of tiny tracers, specially designed radioactive molecules. The PET scanner tracks the position of the tracers inside Dave, revealing his brain function in 3 dimensions. The tracers don't come from a stockroom shelf. Each one has to be made fresh at a nearby laboratory. This is Dr. Miller. He's making the tracer for Dave's PET scan. First, he needs to produce the radioactivity using a device called a cyclotron. It smashes high-speed particles into ordinary atoms, transforming them into new radioactive atoms. Now the clock is ticking. The new atoms are giving off their radioactivity, becoming plain old regular atoms again. There goes one. It means Dr. Miller and his team have to work quickly to build the tracer, a biological molecule. This is what makes PET so unique. The molecule could be anything: a sugar, a protein, a hormone. Each one is tailored to a specific function in the body and will show doctors where in the body that function is happening. Dr. Miller's team runs special chemical reactions to create a tracer from a drug used in the brain. The new tracer must pass quality control. It is purified and checked that it will function properly. The tracer is ready to use. Good work, Dr. Miller. The precious custom-made molecule is rushed to the PET clinic. Just the right level of radioactivity is measured out. Then it's injected into Dave's bloodstream. There'll be a sharp scratch. It's not long before Dave's brain has absorbed the tracer, and the clever chemistry gets to work. Inside Dave, an incredible reaction is taking place. The radioactive atom on the tracer loses its radioactivity, giving off a subatomic particle called a positron. It's just like the tiny negatively charged electrons found on all atoms, except that the positron is positively charged. So when the positron hits an electron at just the right speed, they combine and destroy each other. The energy is released as 2 gamma rays that travel straight out of Dave's body in opposite directions. The PET scanner is like a big ring of Geiger counters. When it detects 2 gamma rays on opposite sides of the ring, the doctors can calculate where in Dave's body the tracer must be. By detecting thousands of these events every second, and with some clever computer processing, Dave's brain activity is revealed in 3 dimensions. Something no other imaging technique can do. For Dave, it was just a small injection. That wasn't so bad, huh? All made possible by the incredible technology of the tracer. The potential for PET is enormous. Different tracers can detect cancer, test heart function, and even track Alzheimer's. The PET scan has revealed Dave's brain is functioning as it should. Dave, you're A-OK. In the next hour or so, his radioactivity levels will be back to normal. It's another successful job for the incredible tracers. But the work of Dr. Miller goes on as he continues creating new tracers to help those in need.
Okay, so here we learn that the key to PET imaging is this radioactive labeling. of some compound that's familiar to your body, like glucose or water. And we then administer the radioactive material to, in the video, it was a patient, but in a research setting, we would be administering it to your research subject or participant. And in this video, you saw it injected, you saw it administered via injection, but it also could be administered in other ways, for example, by inhalation. And what PET images is the electromagnetic radiation induced by the decay of the PET radioisotopes. So you need to choose radioactive material that has a short half-life so that your research subject doesn't run around radioactive for a long time after the experimental session. So that's important. And as you saw in this video, the PET radioisotopes then emit a positron, so a positively charged electron, in the process of decay. And when this positron collides with an electron, the two particles annihilate each other, and they produce these two photons that travel in opposite directions. And that then induces electromagnetic radiation, which is detected externally during the PET imaging, and it's used to measure both the quantity and the location of the positron emitters. This is how PET allows us to look inside the brain and to detect brain function. In PET imaging, the dependent measure, so the thing that we're measuring, is called regional cerebral blood flow, so RCBF. That's the acronym that you'll see in articles that report on PET research. The spatial resolution of PET is very good, so it's about 4 millimeters throughout the brain. But the temporal resolution of PET is very bad, so something like 30 to 40 seconds. And when I say very bad, I mean very bad for the purposes of imaging language processing. So language processing is very fast. The relevant time scale really is the time scale of milliseconds. You can imagine how quickly like a single syllable passes by. It's like 200 milliseconds, and there's a lot of sub-computations that already go into that. So for the purposes of imaging the temporal unfolding of something like language, this system isn't really, this measurement technique really isn't able to do that. The lack of temporal resolution also has some consequences for the types of experiments that we can run with this technique. So for example, you couldn't have an experiment where the types of stimuli that you present to your participant change at each trial, so that you would have to then kind of separate the trials from each other during the analysis. And that's because the temporal resolution is so slow. So you'd have to wait a long time before you present the next stimulus in order to be able to separate the elicited activities in time. So that's a limitation. And for that reason, PET imaging generally uses a so-called blocked design. And by that, I mean that the participant receives just a single type of stimulus. So stimulus comes from one kind of category many, many times within a block, and then in the next block, the stimulus type changes. And that's a major constraint on the types of experimental designs we're able to use with our research participants. So what are some of the pros and cons of PET imaging? Well, the pros have very much to do with the good spatial resolution. And when we compare PET to the technique that we'll talk about next, fMRI, there are certain brain regions that PET images better than fMRI. So in particular, anterior regions of the brain. So those are areas that are a little bit tricky for fMRI. So that could be a reason why you might want to use PET instead of fMRI if you're specifically interested in these anterior regions. What are some of the cons? Well, the main con is that although we're currently discussing non-invasive functional neuroimaging techniques, PET is to some extent invasive, obviously. So it involves this injection of radioactivity into the participant. So that's, of course, invasive. And because of that, there are many limitations to the kinds of participants that you can have in a PET study. So for example, you wouldn't be able to use a person that's pregnant. This is really not that great for kids. Basically, any kind of vulnerable population, PET may not be ideal for. You also can't do lots of PET studies, so there's a limit to how much radioactivity you want to subject yourself to. And because of these, because of the slightly invasive aspect, nowadays, if you want to get human subjects research for a PET study, you kind of have to show that your research could not be used, it could not be done with a less invasive method like fMRI, for example. So it has to be kind of the only technique that will do the job. And for that reason, you see fewer and fewer PET studies in the literature these days. So it's basically been heavily replaced by fMRI. And I didn't mention the earlier point here. So also, like, basically no useful temporal resolution. So you just get spatial information. And so next we'll talk about fMRI.